Okay, welcome back to the final part of this demonstration for the 16 drug cut from UK Drug Testing and All Test. If I just show you, this has got a urine sample in it, and if I just bring that close, you'll see the green pad next to the 3696 above 36 degrees centigrade on the bottom is the one that's showing. So the temperature check on this urine sample is absolutely fine. What we're now going to do is use the colour card just to evaluate what we've got on the adulteration test strip as well. So you look for the abbreviations, look for the matching negative reading that you'd expect to find for nitrates, and that is normal. You then will go to glutarate and test for that, and basically work your way through the different adulterants to make sure that the colour change showing on those pads is all matching the negative or normal levels that you would expect. And then this urine test has now just been running for approximately 2 minutes. But you can see, if I bring that into focus on the camera, what we've got already showing along the top there is a full set of control lines all the way along, with underneath it a set of developing test lines. Now what you've got to do here is wait for the full development time of this drug test before you interpret any positive results. A positive indicator is shown as a control line in the absence of a test line, so no test line forming at the read time for the test, which is between 5 and 10 minutes. Now, if you do get all negative results showing in a shorter period than that, you can move on to the next test, because these test lines, once they're formed, are not going to fade or disappear. But if they haven't formed, and if we show you around here on the pregabalin one, we've got a line just very, very slowly forming, but that's the most resistance to showing a negative result here, and the line is very, very thin and very faint at the moment. So in that situation, if there's any doubt, we would say to the examiner to wait for the full test time to develop, and then read the result that is showing at the correct read time. And then after that, you do not come back to it and look if any other lines have formed beyond that. The correct result is the result that is showing at the read time for that drug group. So what I'm going to do with that is now leave it for a full 5 to 10 minutes, and then we will interpret the results that we've got from that urine sample.